How often do we pause to consider the increasing problem of insecticide resistance in malaria control? This growing issue is of particular concern in regions where the battle against malaria is a part of daily life. In the Arjo-Didessa area, for instance, a study was conducted to assess the susceptibility of Anopheles mosquitoes to existing and new insecticides. This region, known for its sugarcane plantation, is an area where insecticides, pesticides, and herbicides are used intensively. The methods of this study were indeed meticulous. Adult Anopheles mosquitoes were reared from larval and pupal collections of the Arjo-Didessa area and its surroundings. Subsequently, these mosquitoes were tested for their susceptibility to selected insecticides. The samples that did not survive and those that did were identified to species using species-specific polymerase chain reaction, also known as PCR. They were also analyzed for the presence of knockdown resistance alleles using allele-specific PCR. The results of this study were enlightening. Out of the 214 Anopheles mosquito samples analyzed, 89% were Anopheles amharicus and 9% were Anopheles arabiensis. The mortality rates of these mosquitoes exposed to deltamethrin and alpha-cypermethrin were 85% and 86.8% respectively. Mortalities against permafos methyl, bendiocarb, propoxure, and clothianidin were all at or near 100%. Interestingly, none of the subsamples examined for the presence of the knockdown resistance gene carried the L1014F, the West African allelic mutation. These findings indicate that Anopheles amharicus and Anopheles arabiensis from the Arjo-Didessa sugarcane irrigation area are resistant to pyrethroids. This resistance could be synergized by the extensive use of agricultural chemicals in the area. The occurrence of pyrethroid-resistant malaria vectors could challenge the ongoing malaria control and elimination program in the area unless resistance management strategies are implemented. Given the resistance of Anopheles amharicus to pyrethroids, its behavior and vectorial capacity should be further investigated. The study has opened up a new understanding of the complexities of insecticide resistance in malaria control, reminding us of the need for constant vigilance and adaptation in our fight against this relentless disease.